Hi class, Dr. Jim here. In this lecture we're going to be looking at the survey of the prokaryotes, and earlier on in this semester we actually looked at what a prokaryote was. And a prokaryote is a cell that doesn't have a nucleus and it's lacking all membranous organelles, or really any organelles. So we're talking about one of the simplest organisms that are out there, and these guys are still around today. We have lots and lots of different types of prokaryotes, and we're going to be looking really at three main types. The first type is the bacteria, which you kind of associate with most prokaryotes. We're going to talk a little bit about the cyanobacteria. Were once classified as algae, but now are classified as bacteria. And then finally we're going to look at the archaebacteria. And the archaebacteria are the ones that are kind of the weirdo bacteria. They're found in very extreme environments. And then they're not really the bacteria because they're not real true prokaryotes or true bacteria, and they're not eukaryotes. They have some tendencies that kind of lead them more into the eukaryotic range, and so we're going to take a look at these as well. And so that's what we're going to look at today, is all these different types of prokaryotes, or all these different cells that make up the prokaryotes, and kind of go along from there. So let's take a look at this lecture and see. So the first thing is we're going to find out what are the prokaryotes, and we've talked about this before. Again, these are the cells that lack the nucleus and organelles, and the other cool thing or interesting thing about them is that they have exponential growth through binary fission, and so they replicate very quickly, and they can divide into millions and millions of cells actually very rapidly, and I'll kind of give you an example of that in just a few minutes. We'll look at some of the different types of bacteria, so we're talking about the true bacteria, and we'll look at their cell walls, their shape, their oxygen requirements, and then find out where they are. Some of them are free-living and others are symbiotic. So some of them have a commensal relationship in which they benefit, but they really don't do anything to us. Some of it's mutualistic, where we both benefit in the relationship. And then obviously the last one is when you have a parasite or when you have an infection. And we'll talk about all three situations with that. Then we'll look into how do bacteria transfer their DNA, because this is really important. This is how we worry about bacteria actually exchanging their DNA and getting resistance, and we'll talk a little bit about that. They can actually exchange their DNA and get genes from different bacteria, and we'll look at that. Finally, we'll look at what are the cyanobacteria, and I kind of alluded to those are the original blue-green algae, but now they're actually considered bacteria when you look at them. And then finally we'll talk about the Archaea or Archaebacteria, and these guys, like I said, are the extreme bacteria. We find these in extreme environments, and we'll look at what these guys are as well. So let's go on and find out about the prokaryotes. And so the prokaryotes include both the bacteria, the cyanobacteria, and the archaea, which are fully functioning cells. So we had the video on viruses, and hopefully you've seen that already. And remember that what distinguishes a virus from a bacteria is that a virus is only protein and DNA or RNA, whereas in a bacteria cell, you actually have the membrane, you have the DNA, you have the cytoplasm, you have the ribosomes. And the biggest thing is the metabolism that the bacteria have, and the able to grow and reproduce on their own. They don't need any other help from other cells to do those things. Even though they live all over us and they grow all over us, these are free functioning cells. And so we really call these really the first organisms around, and not the viruses. Okay? Bacteria are very small, and they range anywhere from 1 to 10 micrometers in length and about 0.7 to 1.5 micrometers in width. And so these are very small, but not nearly as small as the viruses. So if you remember last video with the viruses were in the nanometer range, you know, anywhere from 10 to 100 nanometers. Very, very tiny, 10 to the minus 9. These guys are about 10 to the minus 6. So they're about 1,000 times smaller than the millimeter in that range. And again, they're about 100 to 1,000 times smaller than our average eukaryotic cell. They're kind of at the extreme limits of where our microscopes can see, and that. And again, we can see these in the lab, and so you will be looking at these in the lab this week as well. And again, they're found almost everywhere, and that's what makes bacteria flourish, is that they're found in air, water, dirt, anywhere you can think of on us and everywhere else. And really the two groups we're going to look at is the eubacteria, or the true bacteria, which are like the E. coli and Staph aureus, and then these archaebacteria, the extreme bacteria. And this includes the bacteria which we talked about before, the Thermus aquaticus, the enzyme that helps make polymerase, the Taq polymerase, and that stuff. And these are the extreme bacteria, and again, they can grow in extreme environments. So we're going to take a look at both of these things. So prokaryotic structure, again, all prokaryotes lack the membrane-bounded nucleus. And so really the DNA kind of floats around inside the cell. So I always like to describe a bacteria as a bag of water with some DNA and ribosomes floating around in the middle. And they do call the DNA where it is in the cell the nucleoid region, but it's not a true nucleus. There's no membrane around it. It does have an outer cell wall, and we'll talk a little bit more about that in a few minutes. It has some move by flagella, some have pili, some have fimbriae, depending, but not all bacteria move or get around. Some kind of stay stationary, like Staphylococcus, where they kind of stay where they are and they move by dust and other things like that. They all lack membranous organelles, so they don't have any membranous organelles or the little machines inside. And many have these rings of plasmids. And I know before I kind of alluded to what I like to think plasmids are things that allow you to have a better life. And so in my micro classes, when I teach microbiology, I always describe plasmids as the iPod or cell phone that bacteria have. They don't make their lives they don't really are necessary for their lives, but they make their lives a lot easier to do. So they encode for genes that allow them to survive in different areas that they wouldn't normally survive in. So it kind of makes their lives a little bit easier. And so that's what a plasmid is. It's a little bit of DNA encodes for some genes that allow them to live in a better or in more environments and things like this. Okay, so reproduction in prokaryotes is asexual, and the way they reproduce is by this binary fission. And binary fission is basically going from one cell to two. And it's kind of this very low-level type of mitosis. So we don't talk about, dividing the nucleus or doing any of those things, because in bacteria they don't have the nucleus. So all they do is they replicate their DNA, split them into two cells, and then eventually divide their cells. And so I would call this a very primitive form of mitosis, but it's called binary fission. The interesting thing is because prokaryotes only have one chromosome, mutations come up in bacteria very easily, and that's one of the big things, is that you can get the bacteria to get mutations very quickly, very easily, and that allows them to adapt very fast in an environment because they can replicate very quickly. Okay? And really with this binary fission, bacteria can multiply really quickly. You can see here in the amount of time, in 60 minutes, if you divide every 20 minutes, you actually go from one bacteria to the final, in this case, 8 bacteria. So you can think how quickly the infections can occur and everything else. When we say that bacteria double in their number, we call that their generation time. And like I said, an example is E. coli. E. coli can double in number every 20 to 30 minutes. So they replicate really, really fast. And so if you look at the numbers down here, just in this log curve, you can see that very short, in a short amount of time, you can talk about millions of bacteria just coming from one cell. So why do I bring up this example? Well, the big thing is when we talk about infection. And so a lot of times people get that what is called the 24-hour flu, but it really isn't a flu. What it is it's E. coli or some other bacteria that causes you some severe gastroenteritis. You might get nauseated, you have severe diarrhea for a day, and then it goes away. What this is from someone that doesn't wash their hands or doesn't prepare your food properly, leaves that E. coli in there, and then they're allowed to multiply, and they can multiply really fast. And so what happens, you go for a nice meal, you eat that bacteria not knowing it, and those bacteria multiply within 2 to 3 or 4 hours into thousands to millions of cells, and then you get sick. And so you feel it the night after going out, type of thing. You feel it that night, you're miserable for about 24 hours, and then it clears because your immune system does a really good job, or you get rid of it pretty quickly. And so that's just an example of how fast bacteria can actually multiply. And it's pretty amazing how quickly they can actually double. Now, certain bacteria, not all, can actually form what is kind of like a time capsule, or what I like to call a time capsule. And really what they do is they store their DNA in there when the environments get really bad. So if the conditions are drying out, or there's bad chemicals, or there's toxins or other things that would otherwise kill normal bacteria, these certain bacteria can actually store their DNA in this time capsule and come back at a later date. And that's what these really are. So they basically make these resistant endospores under unfavorable conditions. They protect them from harsh environments. They're extremely resistant to heat and other chemicals. And as soon as the time or the chemicals come back, they can produce new vegetative cells when the conditions improve. And so they make new bacteria cells, and then they grow and they flourish once again. And here's a nice near cool picture where you see here's the normal bacteria cells, and in these green are actually the spores. And these typically make it when you get older bacteria in a media culture or whatever. And so they grow, they can grow from these little pods and come back when the conditions are right again. And so these are the things, this is the 90, you know, when your Lysol or other things say kill 99.9% of the bacteria. This is part of the 0.1% that don't die because these spores are really resistant. And so what can happen is you treat it with chemicals, they make these spores, and then after the chemicals go away, they can come back again. And so they just keep coming back. And so that's a way that bacteria can protect themselves. Now, the gram stain is a way that we differentiate bacteria by their cell wall type. And so if we know how many bacteria out there, they're the most common type of prokaryote. And there are over 9,000 different bacteria that have been at least named. And some have been cultured, many have not been cultured. Okay, and one way we can distinguish bacteria is by the type of cell wall they have. Okay, and most are protected by a cell wall, and those cell walls are made out of peptidoglycan. Now, bacteria are commonly differentiated between what we call a gram stain. And a gram stain basically is looking at what type of cell wall do you have. Do you have a thick cell wall, like in the gram-positive, so this black line represents the gram-positive, the thick cell wall, or do you have a really thin cell wall, like this guy, which is the gram-negative? And typically in the gram-negative, we have the LPS layer on the outside, and so we see that as well. And so they really have almost two membranes on the outside. So when we wash the bacteria and go through the staining process, that thick cell wall can suck up the purple stain. And so the gram-positive end up being purple in color because they think of a sponge and sucking up all the stain when you treat them. And again, you'll see these in lab this week. You'll see the thick cell walls, and those will be purple. Well, in the gram-negative, they stay pink because what happens is even though you treat it with purple, this membrane gets washed away when you do the decolorizer. The decolorizer washes this stuff away, and what you're left with is a colorless bacteria until you counterstain with this pink. And so we can really determine the difference between a gram-positive and gram-negative just by the color that they are. So when we think about gram-positive, we think of purple, thick cell walls. When we think of gram-negative, we think of thin cell walls and pink. And so, again, kind of remember that for the test. The thick cell wall is purple, thin cell wall is pink, and that's what we have with the gram stain. It's a really good way to identify what type of bacteria you're working with. Now, another thing we can use besides looking at the structure of the wall is looking at its shape. And again, there's three main shapes that we deal with, and we've talked about this before with the prokaryotic cells. They're the spiral-shaped bacteria, which are called the spirili. There's the rod-shaped bacteria, which are called the bacilli or bacilli, and then the round-shaped coccibacteria, and those are the round, circular, ball-shaped bacteria. And so these are the three main shapes. Now you can have some blending of the shapes where you can sometimes get these coccobacilli, which kind of look like short little hot dogs and things like that. But really what we want you to know is the three main shapes. And again, you're going to see all three of these today in the lab or when you actually go and do the lab this week. And so make sure you look at all three of them so that you can readily identify. And typically you can just look at the shape and say, aha, I know what this is. Okay? So that's some of the diversity. Now some other things about the bacteria is where do they grow? And some bacteria require oxygen to breathe, others do not, and some can do it with oxygen or out, or grow with or without. And so we look at these different types of bacteria. So the first group of bacteria that we know about are called the obligate aerobes, and these are the guys that need oxygen to breathe. They're just like us. We need oxygen to breathe. If we don't have any oxygen, we die. Same thing with these guys. If they don't have oxygen, they die, and you don't have any bacteria. There's also another group called the obligate anaerobes, and these guys, if they're exposed to oxygen, it's actually toxic to them, and they die. And so you can't expose these things. And there are a number of bacteria out there that don't like oxygen, and so you find these in the deep levels of soil. Sometimes you'll see them in the gut and things like this where they do not tolerate oxygen, so they want to be as far away from oxygen as possible. And then the last group of bacteria are those that can actually utilize oxygen and those that don't. And one of the perfect examples of those is E. coli. E. coli grows really well on a plate with oxygen and can grow really well in your gut without oxygen. And so these guys can go back and forth from either one of those environments. And we call these facultative anaerobes because they can grow with oxygen or without. They do just fine with or without, and they have machinery in their cells that can handle oxygen, and when there is no oxygen, they can make ATP another whole way. And so these guys can survive either way and do it very well. And so we have those three types. Liking oxygen, those are the obligate aerobes, not liking oxygen, which is the obligate anaerobes, and then finally the facultative anaerobes can do with oxygen or without, and so they're kind of in between. Now another thing we look at is where do the bacteria grow? And most bacteria are called hemotrophs or called chemohemotrophs because they have to eat. They don't do photosynthesis, and we'll talk about the bacteria that do here in just a minute. But the chemoheterotrophs are the ones that actually have to eat and take in organic nutrients to survive. Now these bacteria may be free-living, and most bacteria are. Most of them are found in the soil, water, and sometimes in the air. But there are quite a few that are symbiotic. That means that they live with another host somehow, some way. Most of the bacteria, I would say, are commensal, meaning that they live on us, but they don't do us any harm. And this would be an example like staph on our skin or some of the bacteria in our gut. They live there, we provide them a nice warm home, but they don't do anything to us or give us any benefit. There's another type called the mutualistic bacteria, which actually provide a benefit. And some of these bacteria that live in our gut not only help us digest, but they provide us some vitamins and minerals to help us survive and do really well. And then the third type of bacteria are the ones we don't want. And these are the parasites, or the parasitism, which basically means that they cause infection. And obviously, they hurt us, and we don't want these things in them. There are a small group of these out there, and we have to be aware of them. And if you take microbiology, you're going to learn all about the different types of diseases. And on the next slide, I actually show you a number of the bacterial diseases that a lot of people can get. And you can see they range from sexually transmitted diseases to respiratory diseases to skin diseases, sometimes digestive tract, nervous, and really they can affect all our bodies. And our normal flora that normally live there do a really good job of keeping us healthy and surviving, and these are called the probiotics. When we don't have the good bacteria to fight these things off, a lot of times we'll get sick, and a lot of times these things come when we're stressed out and we're not feeling very good to begin with, and so they can lead through it. Now, I'm sure many of you have had a certain type of bacteria. I'm not going to go through them and that stuff, but I'm sure at least you've at one point in time in your life experienced some kind of illness due to bacterial disease. Okay? Now, I mentioned before that bacteria can exchange their DNA, and there's actually 3 ways that bacteria can exchange their DNA from one bacteria to another. And some of it kind of looks like sexual reproduction, and others kind of basically is just taking DNA out of the environment and sucking it up. And so I kind of want to show you an example of each one of how these guys do it. And the reason why bacteria do it is that they can attain more genes to survive in areas where they might not be able to survive on their own. So it's an all idea of getting more genes from the environment. Okay? And so, like I said, there are 3 methods of genetic recombination. The first one is called conjugation, and this is what I like to call bacterial sex. And basically it's associated with a pilus. And so a pilus is a structure that one bacteria has, and it kind of acts like a bacterial penis. And what happens is that the DNA is exchanged from one bacteria through the pilus into another. And so you have a donor and a recipient, and that donor gives the DNA to the recipient, and then the new donor now has the DNA, which it can be passed on to here. And you can see here, this bacteria here is giving its pilus to this other bacteria and is going to exchange some DNA. And after that DNA is exchanged, what you're going to see is now this guy is going to look more like this guy. And so it's a way to exchange DNA from one bacteria to another, and that's called conjugation. Okay? The second one is called transformation, and that's basically taking DNA from the outside world and bringing it into your cells. Now, how does this happen? Typically, there's bacteria that get lysed all the time due to environmental conditions, other things that happen, and the cell breaks down. When it does, it releases its DNA. Normally, you see DNA basically is very tough to break down. And so what happens is DNA gets released from the bacteria cell, and other bacteria cell can actually bring it up, kind of suck it in like a straw, and then or suck it up like a sponge and actually bring it into their nucleus. And we actually use this as a molecular biology technique. And you can see bacteria glowing here, and this is done artificially in the lab, and we can actually put genes that allow things to glow into bacteria by doing that. Another example, remember when I talked about in the molecular biology when we took the insulin gene from humans and put it in bacteria? Well, in order to put that gene in, you actually have to do the process called transformation. And what you do is you put that into a plasmid, the bacteria sucks it up artificially, and then makes that protein that we need. And so that's a really easy, useful way that we utilize it in molecular biology, and that's another way that we do it. The final way is called transduction, and transduction uses a bacteriophage. And so bacteriophages are viruses that infect bacteria, and we talked about this last time. And what they do is they go around infecting other bacteria, and sometimes bacterial DNA actually gets incorporated into the viruses. And if the bacteria that gets infected by this bacteriophage is lucky enough, sometimes it will get bacterial DNA rather than viral DNA. If that's the case, a lot of times you can infect cells and just pass along bacterial genes to another cell without actually making new viruses. And so this is another way that bacteria cells can get new genes, is basically through the process of transduction, which requires a virus and a bacteria cell. So for the test, make sure you know all three. Know conjugation, which I call bacterial sex, is basically two bacteria and a pilus. Transformation, which is basically the uptake of naked DNA by the bacteria cell. And transduction, which is basically one bacteria cell getting infected with a virus and getting DNA from that virus particle. And so that's the three ways that we get transfer. Now, I talked about also that some bacteria can photosynthesize, and those that can photosynthesize are kind of in their special group. We call these the cyanobacteria because they kind of have this blue-green cyan color to them. And so we call these the blue-green, and they originally were called blue-green algae. And we thought they were protists, but actually if you look at them very closely, they're more like bacteria because they don't have a nucleus and they don't have organelles. They have this very primitive looking chloroplast or thylakoid membrane surrounding the bacteria cells, and then again they have the DNA on the inside. They don't have a nucleus. And so that's what caused them to move from the protists, which you'll learn about next week, into the bacteria groupage. And so that's why we put them now in the bacteria, because they don't have that membrane-bound organelle or nucleus and no organelles on the inside. Very primitive chloroplasts in that sense. And so these guys we'll actually look at in the lab live next week. So make sure you look out for those things next week. But they're actually bacteria, not protists. Okay? And then the last group of bacteria that we want you to know is the Archaea. And these are kind of those weirdo bacteria. And I like to call them the weirdo because we find these in very extreme environments. The other thing that's interesting about them is that if you look at their rRNA, their ribosomal RNA, they actually appear more like eukaryotes than they do bacteria itself. And so the other interesting thing is that they don't have peptidoglycan in their cell walls, so they don't normally stain gram-positive or gram-negative. And again, biochemically, they look more like the eukaryotic cells than the bacteria. However, they still don't have a nucleus and they still don't have organelles, so they're not really a eukaryotic cell either. So what we like to call these guys and have to put them in their own group is the Archaea. And so if you remember back to the first week when we talked about the domain structure in biology, we have the bacteria, the Archaea, and the eukarya. And so the Archaea kind of get their own domain because they really don't fit into the bacteria and they really don't fit in the eukarya. And so that's why they get their own little grouping, and that's why they're there. Okay? And again, we find these guys in very extreme environments. So we find them in the salt lakes, we see them in the hot springs, and then we also see them in the really methanogenic swamps and things like that. And so we see these in these very extreme environments that really nothing else can survive, but these guys flourish and flourish very well and do very well in these environments. Okay? So to summarize this, we've taken a quick journey through the prokaryotes, and remember, all prokaryotes don't have a nucleus and they don't have organelles, and we looked at the three types. We have the bacteria, and again, we can separate them based on what type of cell wall they have. Typically between the gram-positive, which are purple, they have thick cell walls, and the gram-negative, which are pink and have thin cell walls. We can also look at their shape and remember the three shapes: the coccyx, which are the round, the bacillus, which are the rod, and the sporulum or spiral. They can grow with or without oxygen, so some groups are strictly grow with oxygen, some don't grow with oxygen, and some can do both. Okay. Most of the bacteria are free-living, but we do have some that are symbiotic, and some that even can cause disease, and we've speculated about some of those diseases. And again, there's three methods of how they exchange their DNA from one another, and that includes conjugation, which is kind of what I like to call bacterial sex, transformation, which is the uptake of naked DNA, and transduction, which is the use of a virus. And then we looked at the cyanobacteria very briefly. They used to be called the blue-green algae, but we now see that they're more like bacteria because, again, they lack the nucleus, they lack organelles. but they're very important in producing oxygen. They work like plant cells, make oxygen through photosynthesis, but again, they're more of a bacteria in this sense. And then the last group are the Archaea. These are in their own domain, and again, they're different in the cell wall and biochemical makeup. They're actually more closely related to the eukarya. We find them in the extreme environments. We call these the extreme prokaryotes, and again, they're found in hot springs, salt lakes, swamps. Sometimes you see them in deep thermal vents or volcanoes, and really in the most inhospitable environments, you're going to find these things. And so, again, when we look for these extraterrestrial life on other planets, I bet we'll find these guys first over anything else, because these things can live in many different environments that we don't think that anything could actually live. Okay, so we've come to the end of this survey of prokaryotes. Hopefully you got something out of it. If you have any questions, please feel free to email me or ask me any questions. I'll be happy to answer them for you. I thank you for watching this time, and I'll see you next time.